Welcome to Vision to Venture, the podcast where we talk to leaders in the tech world. In each episode, we chat with entrepreneurs, tech experts, and leaders within the tech space to hear their stories. So, if you're curious about tech, starting a business, or just want to hear inspiring stories, this podcast is for you. Make sure to follow and subscribe to never miss an episode. Let's get started. Welcome to Vision to Venture. Today, we are thrilled to have Eswar Iyer, the CEO and co-founder of Aikium, join us. Aikium is a protein therapeutic startup revolutionizing drug discovery by combining synthetic biology and artificial intelligence to tackle some of the most challenging drug targets. So, hi, welcome to the podcast. How are you?
Hi, Emily. Thank you so much for having me here. I'm doing well. Really excited to speak with you.
Perfect. So it'd be really great to sort of dig into a little bit of your background. So I guess kind of what sparked your interest in synthetic biology and also deep learning? And I guess how did your background actually prepare you for this field?
Yeah, it's been a long journey, and
Yeah.
neither synthetic biology nor deep learning were fields when I was doing my PhD or
Hmm.
doing anything in my undergrad. I think machine learning was evolving, but I always had a fascination towards building things with a rapid design build test and making useful things. And that led to curiosity about biological systems as new engineering principles, and in the process realized that it's non-linear, complex, multimodal, and then started thinking about how do you really tackle this. And in that thought evolution, at some point realized you need to have quantitative readouts. A lot of traditional biology perhaps is done, you know, there's good science that happens, but there's also a lot of arts and crafts that happen,
Hmm.
and that leads to a lot of variability so having some quantitative reproducible way of measuring these biological systems and then perturbing them. And I felt like the algorithms will evolve where we can sort of make sense. So that stream kept coming in mind. I was working on, slowly it evolved from molecular biology to say synthetic biology during my postdoc. PhD was molecular biology and postdoc was synthetic biology. And then I joined a biotech company where I worked on, uh, tools for in the next generations of synthetic biology tools. And somewhere it became very apparent that the time was just ripe, where the synthetic biology tools have come of age, and the deep learning has come of age. And
Hmm.
this is a time to make reason more about proteins, which have been pretty elusive. So it was really trying to chase the problem that these tools became apparent rather than chasing the tools. And it just happens to be really the best tools today, and that may evolve as well.
Yeah, no, that makes sense. So obviously kind of all led you, I guess, to where you are now. And this episode is sponsored by Enigma. Enigma help connect machine learning and software engineering talent with groundbreaking opportunities in the field of AI. So, whether you're a company searching for exceptional tech talent or an individual seeking that perfect role, Enigma is your go-to solution. Thank you to Enigma for sponsoring Vision to Venture. Was there anything particular that led you to actually kind of starting Aikium? And I know obviously you're a co-founder, so how did your sort of co-founders come together and actually build the company from start?
Sure. So I guess I have to give a little bit of a background here, but startup was really not a career path when I was in my undergrad or, my PhD. That was not the most obvious career choice. But I was always, had this entrepreneurial energy, and I wanted to build things rapidly. I was not as interested in just publishing a pretty paper. So my PhD was trying to understand neuroscience from studying single cells, doing technology development.
Yeah.
And then postdoc was trying to build a human brain on a chip for high throughput screening. And the idea
Mm.
there was, how can we sort of really push the drug discovery frontier, and what is the bottleneck? And felt like if you could make tiny human brains or microfluidic chips that are reproducible and quantitative readouts, it would be very powerful. So I was
Yeah.
fortunate to find an opportunity at the Wyss Institute, the School of Engineering, and then with Professor George Church working on this problem. And then there I learned a lot of tools fundamentally on synthetic biology and, some on machine learning. And then that's really not my deep background per se, but I understood how to use these different tools. And then what are the limitations with biological assays? How do you do rapid design build test and so on? And then that led to a job at 10x Genomics, where I was deeply involved in leading some of their spatial efforts. So at some point, it became very clear that machine learning and protein engineering were the next big frontiers that were not solved. It was just an intuition. You kind of have an intuition about something because you're in the field or you've been thinking about it a long time about what is the next big problem. So that became very clear that it was there. And I did not know what the problem was that we wanted to solve exactly. But I knew that the first principle is, and by this time, I think I had also become very, my thinking is a lot drawn by some principles, because it's very hard to make decisions when you have too many parameters. But if you can have some decision-making criteria that simplifies that parameter space.
Yeah.
So when I was not sure what area to do this, I mean, what exactly are we going to do a startup in? I knew it was machine learning and protein engineering. And I knew the first principle was find the best people to work with. Are also deeply passionate, are thinking about some problem or trying to solve the next big problem, and have that energy to want to solve this. So I was very fortunate to meet my co-founder, Shankar. We co-worked together in the past. And then we are looking for a machine learning person, and we're looking for the best machine learning person who can bring this to the table. My co-founder, Shankar, is the protein engineer, and I'm the platform technologist. And we couldn't really find a good person. We had some people that we were speaking with, and we were not as excited. And one day I asked my wife, Srividya, who's the best person in this AI machine learning? And she turned around and said, Venkatesh Mysore. And that was a very obvious answer for her. So then I asked her for the introduction, and then we got connected. Turns out she had worked with Venkatesh Mysore almost a decade back when she was at MIT, and he was at DE Shaw Research, which is a premier place for doing computational work. And he was there for 11 years, and she regarded him very highly. I called him, and we basically hit it off. And he was most recently working at NVIDIA on some very challenging problems. And anybody who wanted to apply the best-in-class machine learning, he would help them apply that as a principal solutions architect. So we really aligned, and then we started reading a lot. And in that process, we had some other principles, like not to worry about, exactly the titles or, the things, or flipping a company. Just what's the big problem we are going to solve? If we have very limited span by which we can be productive, and you could get a job anywhere doing anything, why put everybody through this risk of doing a startup? What is that fundamental problem that you're going to solve? I think in that process, we identified a major elephant in the room, in that just deep reading and reading and reading. And that's really where we felt like, oh, this is a problem where we have a founder market fit, where there's a problem and the founders have the exact technological expertise that is not obvious, that is not common, and we can come together to solve this problem. And if we solve that, that can actually, and if there's a very small probability of success, that's fine, we'll take that. Or very high probability of failure, rather, we'll take that. But that's totally worth it. We had to feel like it was worth it. And that happened to be that currently most technologies cannot hit half the proteome. And if you could just design certain proteins in a certain way to tackle this problem, you can suddenly make more therapies for diseases that are inaccessible. And that felt very appealing to us, because at the end of the day, you're trying to improve human experience for the better. And that's kind of how this whole Aikium came together.
No, that makes sense. And to be fair, you went into it a little bit there in terms of kind of what you guys are doing and how you're actually solving the problems. But you guys have a very specific technology in terms of what you're working on. So how do you guys actually address sort of the gaps or the limitations, I guess, actually in current protein therapeutics? Is there anything that you guys are doing in particular?
Yes. So current therapeutics fall under, I guess, two major categories. You have either small molecules that you can use. These are, small molecule drugs like your, paracetamol or anything else.
Yeah.
Antibodies or protein-based drugs. There are a variety of those. One of the things that really hit us as we dug deeper is proteins exist in two forms, or proteins have two proteins exist in a structured form. These are the beautiful alpha helices, beta sheets. You think of a protein as a beautiful symmetrical structure, but there are regions of the protein that are not structured. They are highly dynamic. They're extremely flexible. And you think of them as loops that connect them and so on. These are ropes that connect them. And you think traditionally the view was these are not really important. The domains are important. It's all about domains. Turns out these flexible regions are extremely important because that's really what makes the domains move around. And those are the regions that are so flexible that they are dynamic. They're involved in protein-protein interactions, protein DNA interactions. For example, transcription factors that are at the core of regulating life from a DNA perspective have all these disordered regions that literally go and modulate this, bind different pockets and then activate these genes. There are kinases that are important, and these disordered regions connect these domains. And when they get phosphorylated, that changes the hydrophobicity, and then that changes the structure of the protein. So as important as they were extremely, they were not really as in the center of what we were understanding. Now it's becoming more center. And the fact is no technology really allowed us to bind these regions or modulate these regions. Antibodies prefer a structured region, just the way they have evolved. Mm-hmm. And if you look at all the antibodies for which we have structure, more than 90% of the antibodies are bind to some structured region. And then you have small molecules. They like a pocket. Given a protein, they look for a pocket and they try to go and fit into a pocket. So we don't have either of them. So we realized that if we could make a protein that can bind in a sequence-specific manner to these disordered regions, that would be a game changer, convert them into structured domains upon binding. So that was really the core technology. So how are we different from others? Well, essentially, we're going after the disordered proteome, which is very important. It is core to, it's present in almost all therapeutic targets. So you're getting a new handle at addressing diseases. So you can go after all the existing targets and new targets. And the second differentiator is, so we're going after, the first one is we're going after regions that other technologies are not going after. That's the first differentiator.
Yeah.
Second differentiator is that we use deep learning and we are good at that. But deep learning algorithms are limited by the data that you train them on. And this insight deeply, more than me, resonated from my co-founder Venkatesh, who again came from NVIDIA atomized D shop. He's like, look, algorithms are only as smart as the data, so we should really get the best quality data. And so we almost bet most of the money that we raised on developing platforms that can generate that data in that throughput. So that means that now we can, and the reason that is important is most of the protein algorithms, structural algorithms, kind of refer to each other, or they refer to a few crystal structures. It's almost like an inverted pyramid. So there's a data sparsity problem. You may have structures of maybe a few hundred antibodies that are binding disordered regions, if that. You really need a lot more data of paired binding interactions so you can build these models better. It really comes down to that massive scale of data that can represent this in a clean, paired, high-quality manner, binding interaction that you can train your models on. So we built novel models and algorithms. We built workflows by which we have a wet lab where we can screen 1 trillion molecules, 1 trillion AI-designed molecules in the wet lab. And then the data is read back, and then we ask which of these molecules that we designed bind, why, and then we can iterate this. So in the process, we are building a data moat, which is going to become very important and valuable. As you can see, algorithm, when ChatGPT was there, took 600 billion. Now we have another algorithm from China that perhaps for an orders of magnitude is perhaps achieving the same, then how do you differentiate it? We have similar problem here. So the data is going to be what we are focusing on, and building therapeutic binders is what we are focusing on. So data moat, going after regions that others are unable to go after, and sort of those would be the two major differentiators that I would say.
Wow, there's a lot going on there. That's very interesting, to be fair. And obviously, you guys have been really successful with that. And I know you guys started as a sort of academic lab, didn't you? So have there been sort of any sort of unique hurdles that you've had to kind of get through by transitioning into actual kind of like a commercial biotech company?
Great question. We actually did not start from an academic. We all have academic training backgrounds, but we all were in industry, solving some hard problems.
Yeah.
So Shankar was designing novel enzymes that are going into new, kits, or solving some very hard problems in that space. Venkat was, scaled up the drug discovery, computational drug discovery process quite a bit in his past, and applied, different techniques like structural techniques and deep learning techniques for understanding difficult proteins. And I have helped build several platforms and architected some platforms that are now commercial. So we have that background, and we came from industry. We had an academic background, then went to industry, and we literally started Aikium from scratch. Like, we didn't have any IP that we could license from academia. It was a new idea. It was sort of very different from what everybody, everybody said, oh, this looks very different and it's too far away. So we had to build everything from scratch, literally. So that was extremely challenging to raise the money, to build the thing. And then, of course, we caught momentum afterwards, but it was not directly from academia. But if the question is, how did the transition from academia, what is the transition you see from academia to industry or sort of building startups? I actually answer that.
Yeah.
The key aspect in academia is, and this may be a biased perspective, but this is my perspective, and academia really rewards and cares about the first author and the last author, maybe the second author a little bit. In that process, so academia is great in that it gives you a space to work on problems that necessarily don't have a commercial outcome immediately. And that's the beauty of academia. So you can really pursue some intellectual curiosity. You can go after things, and 1 in 10,000, if it works out, is good enough. I think it pays for itself in the greater good. 1 in 10,000, 1 in 20,000. That's still a great number. But what happens is, in academia, because of this focus on the last author, when you're trying to do hard problems, like a large, you're trying to have a big team that's trying to solve this, besides just say, small team that's trying to do this in a closed setting, then the incentives are not set up properly for everybody to collaborate and team up. Some people do that still well, and they have amazing models to do it, but it is not always incentivizing the teamwork that you would expect. It still happens. Whereas in industry, and again, industry also has the opposite also, but I'm leaving exceptions. A good industry, that everybody is on the same boat. You either sink or swim together. So everybody's on the team. It really doesn't matter if you're brilliant or not. Can you help the team solve a problem, an important problem? I would even say that perhaps it does not even matter whether you have a degree or not. Degree matters. It's correlated to something, but are you just brilliant at solving a problem? Are you just passionate? Can you solve a problem?
Yeah.
If you're able to solve a problem, then that's totally worth it. Then the whole team advances. So you're thinking more in terms of team rather than just first and last author. Of course, you still get, you recognize for your contributions, but it's much. And the second thing is, it's fast paced. When you go home in the evening, you're not thinking, oh, it was all, what am I doing? In academia, you never have a break. Even on weekends, I felt like I should be working. And because I was very passionate, nobody forced me. I enjoyed it and I was passionate, but there was no break. Whereas in industry, and I won't say this for startup, but in an industry, you can still go home. You can trust that your team is working, on the problem, you've done your part and you pass it on, and now it's a teamwork. You kind of have to develop the trust and you work with people. So that would be the key difference.
Yeah, that makes a lot of sense. It's actually really interesting to hear you discuss that as well. Obviously, from your background, you've experienced a bit of both. So I think a good question for you to answer because of your background would actually be, do you have any sort of specific advice for aspiring sort of biotech entrepreneurs who are currently kind of navigating the early stages of actually building a startup?
Find a problem and solve it is the most simplest one. Find a hard problem and solve it. Sometimes the harder the problem, maybe it's easier, but sometimes maybe there's an opportunity to solve something that you can do this. I'll maybe talk about it the way I think about it. If it's a good idea, then it's usually not a good idea because everybody's thinking about it. And I'll ascribe this quote particularly to one of my mentors, Ed Boyden, who I remember him saying this quote somewhere or reading, but it makes a lot of sense. If you think you have a good idea, it's usually not a good idea because many people think it's a good idea because it's at the surface. You just had an idea and you've not done any more diligence. It's possible that 1 in 10 or 1 in 50 of those ideas is truly brilliant, but you really have to do the diligence for that idea. You can't start, really have to look at it. You have to ask who else has done this. Is there any other IP? Are there companies working on it? When you do this, you realize that none of the things you're thinking are necessarily, people are always thinking about this in different ways. So you have to almost find that fundamental problem that may not be hot now or fancy now, but ask in 5 years, what would that be that's going to be important? That's a bottleneck today and opportunity, and you have to go after it. So that's the first one. So think about it from a very fundamental step rather than being carried away by the things that are fancy. Whatever you're seeing being fancy now was in development for a while, and it was not even visible for a while. So you want to be doing those things that may not be necessarily visible now, but maybe fancy in a couple of years.
They're going to become trendy, aren't like you've got to jump on the trend and the bandwagon, I guess, before it becomes that. So
exactly.
It's really interesting.
And here's where I would say academia is fantastic, because in academia, you can do those things. You have the luxury and liberty to think 5, 10, 20 years ahead in your field. And that's how I would use that. I would sort of do this in academia. I feel like there's a lot of fear and about few things, not just academia and industry too, but the fear of my idea. I want to protect this idea and I don't want to share this with anyone. And I want to sort of be rewarded for this. And it's a reasonable one, but sometimes it can go overboard. You really want to rapidly work and then evaluate your idea. And trying to sort of think ahead and ask fundamentally what the problem is. So PhD trains you for this. So I say PhD is not important, but I'm talking about exceptions. There may be people, I've known people who may not have a PhD, but they're exceptional. So if you're not necessarily exceptional without a PhD, if you don't think that's there's a path to it, you can, work in an academic lab. You can literally dedicate your, several years trying to find a problem and solve that problem. So solving hard problems is what the community rewards us for. Solving hard problems that are meaningful to the community in a more immediate manner. And if you're able to do that, then the community rewards you.
Yeah.
So both of those have to be there. You can't just solve a problem that may not be immediately important. So trying to understand and think about this. The second is the value of a network. Keep building network, get advice. Don't build network just for the sake of building network, but really be curious. Try to learn as much as possible from people. In my case, I really was curious and I wanted to learn about machining, because I was passionate about design build testing rapidly, and I just didn't know how to build things properly. And I found a machinist, an Hungarian machinist who's brilliant. His name is Sandor, during my PhD, and he had a little machine shop, and I bargained that I could help clean the machine shop if he can help teach me. And I was writing a PhD thesis, and it was important enough for me that I took one day in a week, Saturdays or so, to do this. And I learned so much, and that have always paid off in some ways. And he was very generous in teaching me as well. So just if you are curious, just go and learn. People are willing to share. People are willing. If you're sincere enough, someone will give you a shot. People say, "Oh, nobody." The nice thing about our community, and especially the startup community, the Bay Area, and generally, where we are now is people are generous. You have to knock on enough doors.
Yeah.
And if it doesn't happen, it's okay, but you just have to ask the right questions to the right people, and or find, sometimes you may not even know what questions to ask. Just keep trying and then keep asking. So use people and solve hard problems. Prepare yourself to solve hard problems. Maybe learn, if you get some insight on business, learn that, because often the focus is on solving a problem and I'll figure out how it gets applied. And academia also thinks a lot about this. But what about if you ask, what is the opportunity here? How do we, for a startup, right? What is the opportunity here? What is the commercial opportunity here? And that may not be obvious either. See, that exercise sometimes may not be obvious that this is going to lead this, but you can do your best still and ask, what is the big problem here where there's people are trying? You talk with a lot of people and you learn that in the process. Validate it by rapid prototyping. So if you have an idea, try to don't incubate it for too long. Just go build it. If you don't want to build it, find people who can help you build it. Talk to people, and you will find that after talking, if you lose your enthusiasm after 3 conversations, you're probably not going to be excited enough to work on it. You should really feel a burning passion. You should feel like you're prepared. You are the person who have been prepared to do this, and this is a problem, and this is important. And unless you do that, if you're still asking, should I be doing a startup, you're probably not ready to do a startup. It should be compelling enough that you do this, and for that, just fail a lot, just rapidly prototype it, and then keep moving forward. But don't give up too easily either. Sometimes it may not be obvious, right? Then be aware of some of the understanding of regulatory requirements like FDA or related guidelines. Just be more aware of the ecosystem that you're sending your invention into. What does that look like? Who are the key stakeholders that are playing there? Venture capitalists, there's FDA, there's, other players. Where does academia fit in? How does all of this come together? Try to read some books, try to learn as much as you can. And today we are at an age where information is accessible is democratized. You have Chat GPT and many versions of it, or any other things. You can read up whatever you want on the internet. You have YouTube. So there's no excuse for not learning what you want to learn. You can't say, oh, I didn't have access, or I don't, everyone has internet. And finally, resilience is one of the key things. Academia teaches resilience in some ways, but you need a pretty strong resilience when you're doing a startup and you don't know whether you're going to have funding in a month or not, and you're still planning 6 months ahead. You need a certain resilience to have that. You work with people who have certain resilience. So yeah, those are the things that come to mind.
That was a really good answer. I think a lot of people will find that really interesting. So thank you for going obviously into the detail you did about that. That was I think that's going to be very helpful to a lot of people that will be listening to this actually. And just to change ever so slightly, obviously to get back to you guys at Aikium and what you guys are working on. So obviously sort of synthetic biology and also artificial intelligence are kind of rapidly evolving fields kind of worldwide at the moment. So are there any trends that you're kind of most excited about and that you think might come forward in the coming sort of months and year?
Yes, there are a few trends. First, this generative AI in protein design is really coming of age. The big question is, making a new drug still takes, $1 to $2 billion, and takes a while to do this. Okay, so how can any new tool help shave this time off? That's a key question. Everybody should be asking this. Everybody should be focused on that. Where in that process are you reducing it? So in this process, generative AI, I think, has a lot of promise in discovering novel proteins that can bind novel targets that can do novel biology, right? So you can suddenly get new tools that can find, it's almost getting the tweezer to understand, rather than using a clumsy thumb to study something fine, you get tweezers to sort of study biology. At a high level, I would think of it as read-write problem in biology. You have a biological system which is very complex. You want to read what is happening there, and there are tools like next-gen sequencing, imaging, multiplexed imaging, or any of those, and then you want to write into it. writing has many modes. You can tweak the DNA, CRISPR edits, big deal. You can tweak the RNA, you can silence the RNA, or you can silence the protein. So all of these have different regulation, and we just have not understood it. So go and starting to tweak these levels and understanding what's happening in the normal and disease biology, that's huge. So now we are at a cusp, we are literally at the beginning where this trend is happening. Now, this is going to get hyped, and after maybe a year or two, this may be cold for some time. But in that process, you're going to have a lot of development that happens. This is a pretty standard technology curve that happens. And then suddenly you will see in 5 years or so, proteins that are AI designed are coming into clinic, that are suddenly hitting the clinic. The cost has come down rapidly because, all of these other innovations are happening. People are working on how do I reduce toxicity. There are groups that are working on how do I reduce immunogenicity. And suddenly some of these will get democratized, and they're trying to find how can I capture this market. They all realize they are good at certain part. They can't make the whole drug discovery pipeline. So they are going to make these tools available. And this is going to become symbiotic. So slowly you're going to see an evolution. Maybe in 5 years, you'll see, 4 to 5 years, you'll see these hitting the clinic, clinical testing and so on. But before it gets approved, it may still take 8 or 10 years before it's come out. But once it does, it will change our graph forever, right? If you figure out the rules for making safer drugs, essentially it's how do you make safer drugs that are more potent. And for people like me and my co-founders and my team, the question that we care about is how do we make drugs, medicines that are not just for the top 1% of people. It's not, there's no judgment for people, drugs that are very expensive, because it is very expensive to make it. But how do we do it in such a manner that we have a sustainable business and yet make it accessible?
Yeah.
Who needs it? And sort of break that, how do we fundamentally break that? And that would be a good reason for us to exist, rather than just flip a company or do things. Those are also nice and they are all good, but this would really this gives us a sense of purpose in what we want to do. So that's the aspect I'm excited about. How do we make medicines faster, cheaper, better, and safer? Second trend is robotic automation in labs, combining AI with high throughput experimentation. So here I'm seeing robotics have been evolving and coming. The problem with robotics is there's this funny thing. You spend as much time or more time because the robotics break down and it's not flexible as a human person. And these are not standardized yet in biology, right? Like I said, it is arts and crafts. I'm not saying it in a demeaning manner, but there's a lot of art that goes into a good experiment that a person knows how to get it done. And automating it always is not easy. And sometimes it may have variability and you may not understand it and so on. But I think we are getting at an age where AI can reason about it. And you're also having these cloud lab like integrations coming. They're very expensive now, or they were expensive. They come down to be more accessible. Like maybe they're tens of thousands of dollars. But you could imagine small cells that you will have in your lab where you could literally have experiments that are running. There are already open source automations there, but I'm seeing the integration of AI, smart experimental design. So one person can do the experiments of 4 or 5 people, and they're still driving it. They're still at the driver's seat, but they are using AI to help reason, come up with the gaps rapidly, design experiments that are most efficient, cost and time wise, go to the fundamental bottleneck in that space, and then rapidly do some experimentation. So that trend probably in the next 5 to 10 years is an aspect I'm excited about.
Yeah, no, that makes sense. And obviously you've spoken about the trends there. Do you think there are any sort of misconceptions as well when it comes to AI in the biotech space? And what do you think sort of the biggest ones are?
The first misconception is AI can solve everything. If you just throw AI at it, it'll solve it. I think there's a lot more that happens underneath the hood. And it's also a lot of science and art and engineering to get that to work, depending on how you tweak the weights, how you filter the data, what kind of data are you getting, all of those things. So that the common misconception is throw it and it'll somehow work. It's not straightforward. There's a lot more that goes inside. That's the first misconception, I would say. Yeah.
Yeah, no, that makes sense. I think that's a misconception. I think not just in the sort of biotech space, but just AI in the whole is that, AI is going to be used and it can just fix anything. It's just not the case, is it? So, yeah, I think that makes sense. And going on to just sort of another kind of a little bit of a different question, I guess, about you guys and obviously your role with Aikium. So, has your perspective on sort of leadership and your leadership style changed at all since you've founded Aikium?
Um, yes, in many ways it has. Yes and no. I think the core principles of what you see, what I think should be done is being done of sorts. But you're always actively learning from everyone. I'm learning from my co-founders, and I'm learning from the team that I'm working with, and that process always makes you better and different. In some ways, you always know and hear that leadership is about maybe enabling or serving the team, but you kind of see that you really see that in action on what you're doing a lot of things, and you are setting an example. So actually I've been fortunate, to be honest. In some ways I'm just doing all the things that I've been fortunate to have seen with with the mentors that I've had, and I've applied maybe my own twist to it and my own sort of hacks or things that make sense to me. But and I think the biggest lesson here is how much I've learned from my co-founders. I mean, how
Hmm.
you know, unless you go deep dive on a problem, and you are at the verge of something exciting, but at the same time something extremely fearful, because you don't have, sometimes you may not know at early stage whether you can solve this. Do you have the funding? Do you have the right thing? But kind of, you feed off of each other's energy, and in some ways
Yeah.
that I learned a lot from my co-founders as well.
Yeah, that's perfect. And I think, yeah, obviously that comes with it. And you seem like you've obviously found some really good people to work with. You've obviously gone into that in a little bit more detail early on about how that kind of came about. So yeah, I think you've probably got a really good thing going on, which is great. And I guess last question then, it would be great to go into a little bit more detail about you guys. So what's actually next for you and for Akium, and how do you guys see yourself evolving over the next year or two? Is there anything specific?
Yes. um, we have built a platform that we think is very differentiated. We think we are addressing a problem that is very fundamental, and I think it's slowly becoming. It was not perhaps as central when we were starting. We had to convince people, but now it's people see it. It's obvious. It's kind of like, oh, I see what you're doing. I think we want to continue building the momentum.
Hmm.
We're very excited. We have a partnership with one of the top three pharma in by market cap. We're very excited about that. It's confidential, but we kind of want to work on that. We have partnerships with three top hospitals, with people that we are very excited about who bring biology. So we're going to be executing on these and try to have, uh, you know, maybe another partnership of sorts. So trying to just work with the ecosystem to create a new drug. Our goal is, and this may be ambitious, maybe in 10 years, we want to have 100 therapies. We want IKM to enable 100 therapies, because this may sound ridiculous and laughable, but if you take in perspective that we live in era of exponential technologies, and I was very fortunate to teach a class with George Church on this. What takes what took 10 years may not take the next another 10 years too. You're kind of in this graph.
Yeah.
We have made 100 therapies with antibodies in the last 35 years. In the realm of these new AI designed proteins, I think it is reasonable to believe that in the next 10 years, we can make 100 therapies. And I want IKM to be making 100 therapies in the next 10 years. And that can only happen if you are open and you work with people who are equally passionate. Same principle, work with the best people who are passionate. And we have spent a lot of time finding such people. We're very excited about the teams we're working with, and hopefully push some of these to the clinic. So that's our focus is going to be just singular. Become very good at what we do foundationally, that we keep leading that space, so protect that core and keep building on it. Make molecules that matter to people that matter. so that we can together go to the clinic. That's going to be the focus.
Perfect. I think that's a really nice way to sort of wrap it up as well. And yeah, I just wanted to say a massive thank you for actually coming on. It's been really interesting listening to sort of your background, how you've got where you are, and then obviously what you guys are doing over at Aikium. I think it'd be really great to sort of follow and see how you get on. So best of luck with that over, however long it takes to do this. And I'm sure there will always be new goals that you'll be striving to as well. But yeah, thank you so much for coming on the podcast.
Thank you so much, Emily. This is it was great speaking with you, and I'm looking forward to this. So thank you so much for having me on this podcast. Really appreciate it. Thank you.
Perfect. Thank you so much. Thank you for tuning in to this week's episode of Vision to Venture. If you're listening, make sure to give us a 5-star review. And if you're watching, like and subscribe to listen to more inspiring stories from tech leaders.